But a neuron can do all of that and also modify specific connections with other cells. And that's what makes a brain a brain. It gives it more specific things to tweak, more dials to modify, more states in which to store that memory. So the capacity of the brain that's consisting of neurons to store information is vastly greater than the capacity of kidney cells or gut cells as we understand them today.
Hello everyone and welcome back to the Neuroscience and Beyond podcast. I'm your host Villain, and together with my amazing team of graduate students, we're on a mission to make science, especially neuroscience, more accessible and exciting for everyone. Today, we're joined by Professor Nikolay Kukushkin, a biochemist and neuroscientist whose research challenges one of the most fundamental assumptions in neuroscience, that memory belongs exclusively to the brain. Professor Kukushkin takes us on a fascinating journey from sea slugs to human kidney cells, showing that memory-like processes may be a universal property of all cells, not just neurons. We will explore how cells remember experiences, how that shapes their future behavior, and what this might mean for our understanding of disease, intelligence, and even consciousness. We also dive into Professor Kukushkin's upcoming book, One Hand Clapping, where he reflects on the evolution of the human mind and philosophical puzzles of consciousness. So whether you're into neurobiology, philosophy, or just love exploring bold new ideas in science, this episode has something for you. If you enjoy the podcast, be sure to subscribe and leave us a comment below. We always love hearing from you. And now, dear listeners, please enjoy the conversation with Professor Nikolay Kukushkin. Professor Kukushkin, thank you very much for coming to our podcast.
Thank you. It's great to be here.
This is a special podcast, not only because we are going to talk about one of my favorite topic, memory, not only because we have you, an expert in the field, but because it's also the first podcast that we record online. So you're now in New York and I'm sitting here in Göttingen, and I'm pretty sure it's going to be amazing. So Today we are going to talk about your research in the field of memory, and of course about your upcoming book called One Hand Clapping. And as usual, we are going to dive into the challenges in academia in our second kind of part of the podcast. Let us start by telling us what is the focus of your research. And maybe if you can summarize also, how did you end up doing what you're doing right now? As long as I'm familiar, you're a biochemist. So how come a biochemist got interested in memory?
You got right to the heart of the matter, because I think that's what really sets me apart from other people who study memory. I like to talk about how most people in neuroscience come to neuroscience from the top down. Usually you become interested in how your mind works, how your psychology works, and then from there you go one level down and you decide to look into, okay, how does that work on the level of neural circuits, or maybe neurotransmitters, or maybe even molecules inside of brain cells. But I came to neuroscience, to memory, from the totally opposite direction, from the bottom up. Like you said, I'm a biochemist. My interest has always been molecules and cells. And then gradually I became interested in, okay, how can you build up a mind out of those molecules? How can you construct the cells in such a way so that thing would start thinking in the way that I'm thinking? So it's a little bit of a different way to think about it. I think that's different. That's the difference between me and other neuroscientists. But I think that both approaches have value. Reconstructing the brain, the mind from the bottom up, and deconstructing it from the top down. You want to meet somewhere in the middle. That's what neuroscience really is about. So what our research is about is what we call cellular memory. It's the ability of cells, and not just brain cells, but cells in general, cells of our body, every kind of cell in the body, even single cells, microbes, organisms that only have one cell, the ability of a cell as a unit of biology to retain information, to register experiences, to have those experiences leave marks on the cell that last for a long time, and then those marks feed back on how the cell operates in the future. So, one way to put what memory is memory is from the past, but it is for the future. And that is true for not just our brains and our behavior as animals, where we go, what we eat, but for the behavior of individual cells. Each cell, even if it's a kidney cell or a gut cell or a bone cell, it has its own experience. It has molecules passing through it, liquids traveling through that cell, nutrients, waves of hormones, signals from other cells, and it's unpredictable and chaotic. And at every moment of time, it's barraged with hundreds of signals, and it has to make sense of them. It has to distill what is important about them and what should leave an imprint in that cell for the future. And that's what we call cellular memory. And what our research shows is that cellular memory, this ability of any cell to register patterns of experience and leave within itself lasting imprints of those experiences, the way that a cell does that is the same way in which our brain cells register our experiences, what we call, normal memory, what we experience as memory, what happens in our brains when we respond to our experiences, when we learn something, when we form a memory. It is the same biological process that happens in each of our cells when they are confronted with their experiences, gut cell experiences or kidney cell experiences. So it's this continuity between different forms of memory, including brain memory, but not limited to that brain memory. That is the focus of our research.
So you dropped a lot of terms, memory, experiences, different cells. I think we're going to dissect everything really carefully, because I truly believe that memory is something very special. It's not only about what we think about the past, where we were in yesterday, etc., but it's a fundamental property that we need to build up reality, if you wish.
Of course.
We can talk about this really carefully going into this. But before we go there, how could you do this? How could you do this? How could you study all of these cells? What kind of organisms do you use to study them?
Mm-hmm. Well, so my research journey in neuroscience started from actually sea slugs. That's what Tom Caru's lab, where I'm a senior scientist, has studied for decades. That's been our research model for many years. Why sea slugs? Because they have very simple memories. They have a very simple nervous system compared to ours, much, much fewer neurons. And you can understand their behavior, what happens in the everyday life of a sea slug, by understanding the communication between, let's say, a handful of cells, not as many as you need to understand in a mouse or a human. And from there, we went one step further. We asked, okay, if you only need this simple brain with only a couple neurons connected to each other, and you can see the changes that are relevant to the sea slug in the modifications of a couple of molecules in those cells. Well, maybe we don't even need those cells to be neurons. Maybe those same modifications would also happen in other cells, in generic cells, if you find the right conditions, if you find the right way to train them in the way that we used to train sea slugs. The way that we used to train sea slugs is by giving them electric shock. This electric shock causes this release of serotonin, which in ourselves it's sort of a happiness molecule, and sea slugs it's more of a danger signal. But anyway, the serotonin gets sprayed throughout their nervous system, and it causes in the neurons of the sea slug a couple of biochemical processes. We can artificially induce those same biochemical processes in any cell we want. And those cells can be, well, in our case we took human neuroblastoma cells, which are not quite neurons, but they could become neurons. They're precursors of neurons. So we thought that by using that cell type, we can ask what is different between neurons and not neurons. So that was the one cell type that we used. And another cell type that we used was kidney cells. not for any particular reason of them being inside of a kidney, but simply because they're not neurons. We wanted to really test if the processes that we're studying, this cellular memory, is a feature really of any cell, not of a brain cell or even a proto-brain cell, but really of any kind of cell in the body. So that's why we use these two cell lines, human cell lines that derived from human cancers, actually, which is why they can divide indefinitely. We can treat them as essentially microorganisms grown in a petri dish, but they are human cells. What they represent to us is generic human cell. It doesn't matter that it's from one particular organ or another. It is a generic human cell. And what we do with those cells is modeled after those experiments we used to do on sea slugs. Basically, we reach into those human cells and we press the same buttons as are pressed inside sea slug neurons when you train them with those electric shocks. We artificially do that in those generic human cells. So what does that mean, for real life? We do that artificially, so what does that correspond to in the cells of our body in real life? Well, it could be hormonal signals, for example. Lots of those biochemical changes that in a sea slug might come from a neurotransmitter might happen in a kidney cell as a result of a hormone coming from another part of the body. It might happen in a gut cell as a result of us consuming food. Lots of different kinds of signals that cells of our body receive all converge on the same biochemical mechanisms. And by turning on those biochemical mechanisms artificially, we're just basically getting access to a large variety of processes that might happen with our cells in real life in our body. So that's what we do with those cells. We artificially stimulate them in the way that we would stimulate a neuron in a brain. And then what do we watch for in those cells? It's also artificial. We have this, what we call a reporter protein installed in those cells. What it means is that when those cells turn on memory genes, we know that they're memory genes because the same genes are turned on in neurons when we form memories. That's well known from neuroscience. We know what kinds of genes get turned on in neurons when a human learns, or a sea slug learns, or any other animal learns. We can take those genes, we can find them in these kidney cells and generic cells, and then we can create this reporter protein that will be produced every time that gene is turned on. So we can't directly ask, is the gene turned on or not, but we can see that reported protein because it's glowing. It's actually taken from a firefly. It's called luciferase. So it glows. It literally glows when those cells turn on the same genes that neurons turn on when they form memory. So we press the same buttons as we press in neurons when an animal is learning. And we watch for the same changes that happen in neurons when memories are formed. We just do that not in a brain, not in a brain cell, but in these petri dishes of generic cells that are taken from the human body. And what we see is that, well, the conversion of one to another, that's what memory really is how you convert the inputs, the stimuli, into long-term outputs, what changes with those cells over time. So this conversion is the same, whether it's happening in a brain or whether it's happening in those kidney cells. What we specifically looked at is this well-known property of memory. It's called the mass-spaced effect. So that's been known about memory in humans and animals. Any system that you look at, any brain will have this property called the mass-spaced effect. If you have the same amount of training, learning, studying for an exam, you study for the same amount of time. If you do it all in one go, if you cram that learning into one session, you will form a worse memory than if you space it in time. Even though you study for the same amount of time, if you space it with intervals, your memory will be superior. So that's true for humans, for sea slugs, for mice, for any kind of memory that you look at. But that's never been seen outside the brain, in cells like kidney cells. And what we have shown by giving them these artificial stimuli, by stimulating their molecules in an artificial way and then watching their memory genes get turned on by watching this reporter gene, what we've shown is that they also have this mass-spaced effect the same amount of chemical stimulation delivered in one go produces less activation of the memory gene than same amount of training just delivered in spaced intervals. So the difference is really just a few minutes. It's a few minutes of how you distribute that chemical signal, but the cells are able to tell the difference and respond to one protocol in one way and to another protocol in another way, just like our brains do to spaced versus mass stimulation. And we also show that the biochemical components of this response are the same as they are in neurons. So the same process is run by the same molecules in generic cells as what's happening during memory formation in the brain. So we think that that's evidence enough to say that it's all one process. It's not like memory. It's not similar to memory. It is that same process. Brains have it, kidney cells have it, all cells have it. It's much more fundamental than the distinction between brains and non-brains.
So first of all, this is incredible. And so I, of course, checked your publication. It was published in Nature Communication, one of very prestigious journals. And I think as of last week, there were like 141,000 downloads of this publication, which is awesome. And what I find particularly cool about it is that you're using a very simple organism and very elegant techniques, and you kind of discover something so fundamental. It's this paper is a huge distinguishment from the majority of papers in these crazy journals that are filled with so many technologies, so many methods, so complicated, and at the end there is nothing novel to the field. That I find particularly nice about your work.
Thank you. I really appreciate you saying that.
Really. Let me comment on the model organism, the sea slugs. This is a legendary organism, the Aplysia, right? So, yes, Eric Kandel worked on this organism way back, one of the fathers of neuroscience, if you wish, or founders of the modern neuroscience, right? And since then, it's been an awesome tool to really like poke neurons that you want to poke exactly and to see exactly what is going on. And as a person who is also working with cell cultures, I really appreciate work like this. But I have plenty of questions about the things that you've said so far. So when you say memory is formed, we have these genes, memory genes, we have proteins running around, etc. Where exactly is memory stored or information stored? Is it in the gene? Is it in a particular protein? Is it in a phosphorylation pattern? Where can we find this information?
I think that the correct answer to be here is to say everywhere and nowhere in particular. It is in the very process of conducting that signal, in the entire cascade of that conducts this information, that the information is stored. It doesn't modify just one point or another point. It modifies everything. It modifies the entire state of a cell, and that it will include phosphorylation of proteins, turning some proteins on, some proteins off, turning on genes, those genes producing products that then go on and modify proteins again, that then go on and turn on other genes. It's loops and feedbacks, positive feedbacks and negative feedbacks, and collectively they modify the cell or an ensemble of cells for a long time, for a longer time than it took you to perturb them in this way. And for as long as that state is modified, we call that a memory. And when it comes back to its natural homeostatic state, we say that the memory is gone. And it might be that some part of that homeostasis has returned to the baseline, but some other part has not returned to the baseline. So it might seem that there is no memory. For example, if we're talking about a behavioral memory, about an animal finding, an escape from a maze, it might seem that the memory is gone, but you retrain the animal and the memory comes back faster and stronger. So it was there somewhere. Something was still modified. It just wasn't immediately obvious. So I think it's wrong to think of memory as something that has a particular place and that sits in there and When it's there, the memory is present, and when it's gone, it's gone. We need to think of the entire brain as consisting of these moving parts, and every part can move in different directions for different durations of time. And when it moves somewhere, well, the brain thinks differently. And for the time that it thinks differently, we say that that's a memory.
Mm-hmm. That's true. I agree to that. But it's very interesting. So when we look at the brain, we have billions of neurons, right? And they're astonishingly interconnected with trillions of synapses. Synapses meaning the connection between two cells, right? And some people would argue, yes, we have this modification of particular cell, but this cell then fires to other cells, then network is kind of triggered, and then we have some kind of experience, if you wish. What is the difference to the memory in these other non-neuronal cells? So do they communicate to each other in a similar way that neurons communicate, or the memory is only within the cell and somehow through hormones and different signaling they kind of convey the message to other cells?
That is a great question. And I do think that there is a degree of preservation of information in these signals between non-neural cells. We see that in our work that, for example, the signaling molecules that are secreted from our kidney cells, well, after a particular stimulation, there'll be more of those molecules secreted from that cell, and so other cells in the vicinity would hear that molecule, maybe modify themselves in response as well. So it does exist, the modification of connections, communication between cells, and it's part of the cellular memory. But there is something that does distinguish neurons from non-neural cells. It's not that neurons are generic cells, they are special. And what makes them special is synaptic contacts. And it's not just the fact that those contacts can go into a far away location in the brain and send a message somewhere far away. No, that other cells can do that too. They can send chemical messages far away in the body. What makes neurons special is the specificity of those connections, is the fact that they can convey a signal not just to some cell in that part of the brain, but to one specific cell in that part of the brain, and then to another specific cell in a totally different part of the brain. And those two contacts can change independently of each other. That independent modification of those thousands of connections, specific connections, that is what makes neurons special, because it gives them a lot more degrees of freedom that they can modify. A generic cell can modify its gene expression. It can increase the expression of some genes, but not others. It can increase the production of some hormones, but not other hormones. But it can't increase a release of a hormone to that cell over there, but not to this cell over here. That it can't do. It can only modify it as a whole. But a neuron can do all of that and also modify specific connections with other cells. And that's what makes a brain. It gives it more specific things to tweak, more dials to modify, more states in which to store that memory. So the capacity of the brain that's consisting of neurons to store information is vastly greater than the capacity of kidney cells or gut cells as we understand them today. Maybe we will discover in a couple of years that those cells also communicate through very specific, synaptic contacts, which is haven't discovered yet. But as of now, we don't know of that kind of degree of specificity in cells communicating with each other. That is still something that neurons and brains are very good at. But it's not to say that in principle it doesn't exist in other cells, and that collectively our body cannot store enough information for it to be meaningful for our lives.
Mm-hmm. That is true. And I think now it's a very good point. So we kind of touched upon the cellular mechanisms of a memory, or molecular even, the genetics and the molecules running around. Now you discuss some of the network dynamics, and now we can go a little bit higher to the systems. So, as a humans, when we think of memory, it always comes this episodic memory of the past, or semantic memory, some knowledge, etc., etc. How do neurons integrate this kind of information and store it? And more importantly, how do we access this information, you know?
Yeah, we humans operate on a such high degree of abstraction most of the time that it's very hard for us to wrap our mind around where is it grounded? What are the actual physical experiences that have led to our highly abstract understanding of the world? Like, you can't think of climate change without having the words for climate and change. You can experience temperature, you can experience humidity, you can experience that being different today from yesterday. But to understand the concept of climate changing over hundreds of years, you need to detach yourself from those experiences really, really, really far away and to compress them into these highly abstract units of language. So it can be really difficult to go back and say, well, okay, the idea of climate comes from, our skin experiencing warmth and our skin experiencing coldness, and then neurons are patterning that information into concepts of cold and hot, and then connecting that to the idea of ambient. It's so far away from this direct experience that it's really hard to say what does it boil down to. And I think for me, working with sea slugs was really revelatory in this sense, because sea slugs are a lot simpler and it's easier to understand how they process their patterns of experience. Like, what danger is to them, for example. What danger is to them is it's any signal coming from any pain receptor anywhere in the body, and there's a neuron that collects all those signals from those different parts of the body, and when that fires, well, the firing of that neuron represents the idea of danger. It's as simple as that. There is no further switch that says danger. It's just the fact that all of those different pain signals have converged in one point in the brain. I think that in a more complicated way, but that is also true for how we process, understand, and access these abstract features of reality. It's not that there is some special climate changeness that clicks in our brain when we perceive this concept of climate change. It's just by virtue of having our brain organized in this hierarchical levels, just by virtue of them processing our everyday experience and being positioned in such a way that one neuron responds to a pattern of heat, one neuron responds to a pattern of cold, and then those neurons form part of a greater pattern of cold weather and hot weather, and then those neurons forming part of an even greater pattern of changes in cold weather and changes in hot weather. And by virtue of our brain being organized in this hierarchical system, that we eventually get to such immensely abstract concepts like climate change. It's really hard to wrap your mind around, because what we want to ask is, okay, where in the brain is this, where does this concept of climate change sit? Point to that place in the brain and say this is climate change. But you can't. There is no place. It's all the combination of all of the things that are related to climate change, activated and influencing each other at the same time, that forms this concept in our brain. So that's how I think about it.
That is very interesting, actually. And you mentioned language, right? If it's curious to think about if people who are speaking different languages, do they have different kind of abstraction, or do they have different ways to store this information or not? This is also an interesting topic of research. But so I think you are familiar with Alexander Luria. He's a Russian neurophysiologist. Some people say the father of neurophysiology, right? He wrote this wonderful books, one of which is The Man with the Shattered World. And I think if people are not familiar with what you're talking about, when you read this book, you really are going to realize about so the book is kind of a blend of a case study and a memoir of a soldier who was wounded in the head. And in this book, it's described very nicely how a person with a brain injury struggles to basically construct reality, things that we actually take for granted. You see that they are not actually granted. Our brain helps us reconstruct the whole very reality that we're living in. And that's why I think memory is the key for understanding, our reality and maybe consciousness. This we can touch upon a little bit later. And I completely agree that, as you said about the climate change, that it's hard to pinpoint a particular point in the brain or pattern in the brain. It is just different organization that we haven't figured out yet. And hopefully, who knows, maybe in the future, we're going to figure out. So a question that I wanted to ask you regarding the sea slugs research. So when we think about our memory, let's say of an ice cream that we ate yesterday or a year ago or whatever, it comprises probably of a huge network of neurons and activity. Can we think about a type of memory that a sea slug would have? If you think about that, the system of the sea slug is very limited. It has like a couple of neurons only. What I want to ask is, if we think about our experiences, to what extent animals that are not humans could have this kind of experiences? Although we can never probably measure that, but what is your opinion on that topic?
Well, you have to think about it evolutionarily. You have to think about at what point in the history of life the sea slug and we were the same animal. And that point in time was probably about 600 to 800 million years ago. There was a common ancestor that gave rise to both sea slugs in one direction and humans in another direction. Lived at the bottom of the ocean. It was probably something worm-like, and it had a brain, but a very simple one. And sea slugs stayed more or less with that simple brain, with a few modifications, whereas we blew it out completely out of proportion and added gazillions of other elements to it. So everything that we share with sea slugs is what comes from that bottom-dwelling worm. And everything that we acquired from that point is different. So what is the same? The structure of a neuron is basically the same. Yeah, they're a little bit different. The sea slug neurons are slower, they're bigger, but it's not fundamental difference. It's details that are different. So what you can translate from a sea slug to a human is how individual neurons communicate with each other, how they respond to time patterns, what genes they turn on, what molecules they activate. All of those things are very, very similar between humans and sea slugs. But what is different is the circuits, is what you build out of those neurons, how you connect them to one another, how you construct abstractions out of them, how you use those abstractions to guide behavior of the animal. All of that has evolved after our split into two separate branches of evolution. So I would say that cellular memory is what you can translate between a sea slug and a human. Not the kind of memory that we think about when we close our eyes and think about the past. And that's something that takes a while to grasp when you're working with these sea slugs, because we always anthropomorphize things. It's very hard to unsee another animal as just a simpler version of yourself. But you have to remind yourself that their experience is fundamentally different, that they don't perceive the abstract categories that we perceive. They don't have a visual understanding of the world as we do. Their eyes are very, very primitive. They don't hear what's happening to them. So even based on those things, their picture of the world is totally different from ours, and you can't easily project yourself into the sea slug. I always talk to my students about whether a sea slug feels pain. I'll usually tell them no. that's just that's just remove the questions. But if you really think about it, it's an impossible question to answer because you can't try on the consciousness of a sea slug on yourself or vice versa. You don't have any comparable feeling parts that would be projectable from one organism to another. There is no such thing as pain that means the same thing for a sea slug and for a human. What we include in that word is really specific to us, and you have to keep that in mind when working with animals like that. But it
Yeah.
also gives you a broader perspective on yourself. I think that if you're working with a mouse is still close enough to a human that it can sort of work. You can project yourself onto a mouse and derive meaningful conclusions. When a can't figure out its way out of a maze, you can kind of put yourself in the mind of that mice and probably be correct in sort of imagining what is going through its mind. But you can't do that with a sea slug. You have to just rethink your entire philosophy of what it is to think, or what a mind means if you want to understand what's happening to a sea slug. So it's philosophically, I think, really useful organism to work with. It makes you rethink the fundamentals.
That is correct. And if you work with monkeys, it's even more interesting to to think about their perception and their memories, experiences. We have here quite a big primate center research, and yeah, it's very interesting to see and hear about the things that are they doing. But I've never imagined working with monkeys simply because they're just too close to us, and
Yes. Yeah.
it's very very strange feeling. Even working with mice gives me a little bit chills, and I hope the sea slug is a little bit easier to when you have to dissect and work
Yeah. Yeah.
in this regard. Yeah,
and
you
and you
mentioned
have to and you have to do it
yeah.
you can do it with a sea slug. Funny thing is that you can deconstruct it to various levels of completeness and they would still work. You can remove individual neurons and they will work. If you want, you can preserve some of you can separate the tail from the siphon from the head, and you will poke one or another and the other parts will still move. So you can disassemble it, reassemble it, and all the parts still work together, and it really makes you question, what does it mean to be alive? Like where does an alive slag end and just parts begin? There is no line. It's totally up to you. It's just how you define the transition.
Yeah, indeed, it's a very interesting question, and also philosophical what life is, right? We
Hmm.
still kind of argue to this day,
Yeah.
are viruses, for example, alive or they're not alive? This is another interesting thing.
Yeah.
But you mentioned translation, and I wanted to ask you, how do you see the findings about non-neuronal cells having this kind of memory in terms of translating them for kind of some, let's say, treatment or procedure helping solve a certain disease?
Yeah. Well, let's be clear. At this point, we know very little about what this cellular memory actually does in real life. What we've established is a proof of principle that it does exist, that cells are capable of it, and they do it in the same way as neurons do it. But what exactly do they memorize in our everyday life? We still don't know. Everything that we're doing so far is artificial. We're giving them artificial signals and we're watching how they change in response. But it opens up the door to a whole new way of thinking about medicine, I think. For instance, we already know that cells in the pancreas, the cells that produce insulin and other hormones, that they change in response to a meal. If you load up those cells with a lot of sugar, it's like you've had a big meal, they release insulin, which helps us absorb that sugar, and then you wait, say 20 minutes, and then you repeat that. You give this load of sugar once again, and now the release of insulin would be doubled. Now the cells will release almost double the amount of that insulin. that's also a memory. They modify themselves in response to this barrage of nutrients so as to absorb them better next time. If they were permanently releasing insulin at this high level, we would probably be fatigued and hungry all the time. So it makes sense for those cells to change themselves in response to experience. That's been known for decades. It's not news. It's just that nobody has thought about this as literal memory or as being connected in any way to what's happening in our brains when we study for class. We know that there's a patterning of exercise that affects the growth of muscle cells, the stability of bone cells. It's just we have not conceptualized it as memory, and we've never studied, okay, what are the exact time intervals where you need to stimulate a muscle cell or a bone cell to achieve, a stronger muscle or a stronger bone. And now we have a way of doing that. So that might have implications for health, like exercise, diet, anything we do in a schedule. It also has implications for disease. For example, when we're treating cancer, our default assumption is give the patient chemotherapy for as long as they can handle it. Maximize the amount of this drug for as long as the patient can survive it. We want to kill the cancer cells, but not kill the human. Maybe there is a better pattern. Maybe we can train the cancer cells to be more sensitive to those drugs, and there is research in that direction already, not necessarily from our lab. Mm-hmm. So that's one direction, thinking of our body more in the way that we think about our brain. It's not a surprise to us that by repeating something regularly, you get better at it. But maybe we can think about our body as learning in response to patterns in the way that we're used to thinking about the brain. But another side to it is that these changes that we're studying in the cells of the body, and kidney cells and neuroblastoma cells, we're assuming that they're also happening in brain cells, that all those same processes are happening in both cellular memory and neural memory. It's all the same process. And so what we think is that we can use the insights, what we know about cellular memory, to better diagnose mental health conditions, for instance. We know that some of these biochemical changes that we study are dysregulated in schizophrenia, in neurodegenerative diseases, in anxiety, and some of them can be detectable in blood, for example, in peripheral tissues. So it might be that we'll be able to better understand what is happening in the brain by taking samples of the blood, for instance, or diagnosing a patient independently of their reporting, which is always a difficulty in psychiatry. You're relying on self-reporting. You're relying on people telling you what they're experiencing and how they feel, and that makes everything more complicated. But if we can have tests based on cell signaling rather than the patient reporting what their mental state is, well, that's a huge advancement in psychiatry. So those are the two directions where we think our research might go. One is thinking about the body more like about the brain, and another one is thinking of the brain more in connection to the body, and just recognizing that the processes that are happening in both the brain and the body, they are related. They are really part of the same thing, and we can get insights into our one or another by looking at the other part of it.
Yeah, so would you think then that if you have a disturbed memory, let's say in the case of schizophrenia, if you have some kind of a disturbed memory in this patient, for example, when you study kidney cells, you could observe some disturbance in this cellular memory pattern. So this is the presumption, the hypothesis.
And that's exactly what I'm saying. We don't know that yet. We don't have such evidence yet, but I think it's a viable hypothesis that really hasn't been explored in great detail. We do have some information about how big swings in cellular signaling, when it's really off, you can detect that in the blood. But we know very little of what you just said. If a person has schizophrenia and has neural symptoms, Does that mean that there are similar symptoms in kidney cells? If a person is more intelligent, for example, if somebody is better at temporal discrimination, for example, there's correlations between a person's ability for person's capacity for causal reasoning, for how we understand cause and effect, and our ability to distinguish between time patterns. So your ability to order events in time relates to your ability to establish causal links between
Mm-hmm.
events.
Mm-hmm.
Well,
Mm-hmm.
and so if our ability to establish causal links relates to our ability to tell time, maybe our ability to tell time also relates to how cells tell time patterns. And maybe people who are better at causal reasoning, maybe their kidney cells are also better at time pattern discrimination, the kind of time pattern discrimination that we study in our experiments. All of these are pretty wild hypotheses, but I think they're not unreasonable. I think this is something that is worth investigating, because it might give us access to new biomarkers, new ways of diagnosing diseases, and maybe even treat those diseases.
Mm-hmm. Mm-hmm. That is true. It would be cool actually if I don't know if people already differentiate kidney cells from patients, iPSCs. I don't know if this is done, but if you can try it out, in these cells and
Yeah,
see, oh, is there any disturbance? Wow, that's incredible. Let's check it out.
yeah, we would love
Yeah,
to. We
yeah,
would love to do that. And
yeah, yeah.
another thing that we are hoping to do, we're writing some grants about it, is um, being able to sort our cells based on how they respond to the patterns of stimulation. We
Mhm,
can, because these are cells grown in culture, we can use flow cytometry to separate the cells that respond strongly versus the cells that respond weakly. And we're interested in tracking what makes strong responders different from weak responders. Is there, for example, a gene that would determine that a cell is really good at discriminating a temporal pattern? And can we then take that gene and go to databases of genome-wide associations with cognitive markers or markers of mental health disease, and find some overlap, some correlation in what predicts that a cell is better at timing or smarter, and that the person is healthier or smarter. We don't know that yet, but we are very, very interested in it.
mhm. That would be cool. I wish you a lot of success with the grants and
Thank you, thank you.
yeah, yeah,
Yeah,
yeah.
we need
We're
it. We need a lot of luck in this day and age.
Yes, yes, we will talk about this in the second part of the our conversation. But indeed, it's dynamic, it's changing, it's insecure, and
Yep.
unfortunately, but hey, the science is incredible. Therefore, we're there and still struggling, so to
Yeah,
say.
yeah, yeah. That's the
um,
life of a scientist.
so yeah, exactly. We've been talking now for roughly 46 minutes. And although I would like to talk for another 46 minutes, maybe, about memory, in the remaining time, I'd like to discuss with you your upcoming book, One Hand
Mhm.
Clapping. So, your publisher was so kind to send me the book, and I read it and I enjoy reading it. And it was very nicely written, easy to read. I don't know, I'm a little bit biased because I'm a biologist, so I kind of understand the terms, but I think also for a reader without any experience, it will be very interesting to follow. You have these very cool tiny doodles or like illustration. I don't know,
Mhm.
did you draw them by hand yourself
Yeah,
or?
yeah, yeah, yeah. Yes, yes. This
That's
is
very cool.
all these doodles are um, evolutionarily derived from doodles that I used to draw in my college notes, and just something that I used to do for fun and make things lighter. And they, because my notes were pretty good, I think that the people in my class would copy them, would Xerox them, and those doodles kind of became well known in my class. And then one of my classmates, Ася Kazantsova, who was a popular science writer, well, who was in Russia. She's in exile now, as many of my friends and colleagues. But she's a popular science celebrity, and for her first book that she wrote. She asked me to illustrate it. She wanted that style of cartoons for her book. So that was my first uh, illustrator gig. I started illustrating before I started writing, ironically, even though I really don't know how to draw anything, as you can tell. But I illustrated her couple of books, and then when it came time to write my own, this was just what I already knew and the style that I did. But funny enough, um, after it was already published in Russia, um, somebody dug out this book that my grandfather wrote. My grandfather, who was a chemist, he wrote in the early 90s, and all of us have forgotten about that book. It was sort of just chemistry for beginners, chemistry for everybody, like these little anecdotes and stories from different aspects of chemistry. And somebody dug out this book, and I realized that it has the exact same style of cartoons. So it must have been lodged somewhere in my memory, and we just talked about access. I had no idea that it was there, but clearly it was. So I like also this fact that there's this continuity between my grandfather's book and this one.
Yeah, this is kind of a transgenerational memory epigenetic
Exactly.
modification,
Yes,
right?
yes, yes, something like that. Yeah.
Encoded.
Mhm.
May I ask you about the name of your grandfather?
Yeah, it's Yuri Nikolaevich Kukushkin. Yuri
Yuri
Kukushkin.
Kukushkin.
Yeah.
Yeah, yeah, yeah. Very
Yeah.
interesting. I will check it out.
Yeah,
Is
thank
your
you.
family
Thank you.
like science-based
Academics? Yes,
academics?
very
Yeah.
much so. Yeah, there's also my dad, Vadim Yurievich Kukushkin, um,
Uh,
who's also a chemist. I'm the renegade biologist. Most of
uh,
my family is all chemistry based, so I went really far away and did biology. But yeah, I do have one paper with my dad from when I was just starting out. I was testing some cancer drugs that were produced in his lab. My dad has a couple of books and few publications with my granddad. Unfortunately, my granddad died when I was 10 years old, so we don't have a triple one, but that would have been great.
That's amazing, actually. That's really incredible. I would love this to feel this, to publish with your dad, and that's
Yeah,
crazy.
yeah,
Yeah,
I'm really proud of it, even
yeah,
though I've never known anything else.
yeah,
My whole life has always been academia and grant applications and papers and professors, and that's
yeah.
just what I knew to be, normal life.
Well, it turned out to be great. So, I mean, you have an
Yeah,
impressive
yeah, no,
career. You've
I
been
don't
to,
complain. I don't complain.
yeah, St. Petersburg, then Oxford, Harvard, now
Yeah,
NYU. So,
that's
it's
right.
it's it's crazy. Yeah, yeah,
I've
yeah.
seen a bunch of universities and been lucky enough to, travel across different cultures and see how it's done in many different places. Been very lucky with mentors.
Hmm. It's very interesting that you escaped the boring, easy world of chemistry and you enter the, craziness of biology.
Yeah, well, that's definitely how I see it. I don't know, not to diss my family's work, but to me, biology just has more of a story than chemistry. Sometimes my dad asks me to look at, proofread some introduction to one of his papers, so I do have an eye on what he does. I don't understand much of it. But what strikes me is that all of their papers they go right to the point. They say, well, here's this molecule. It's been studied here and here, but not here and here. And now we're going to study it here and here. And then we go right into the meat of the paper. There's nothing else. Whereas in biology, it was like, well, 800 million years
Mhm.
ago, there was once an animal, and everything is like this. There's always this story that if you want, you can take to the origin of life, and then from there it continues logically, consistently through some twists and turns of cause and effect to every single thing that you be studying. So
Yeah,
there's
yeah.
just more drama, more narrative in biology, even though it's much messier and I think harder to work with, just because everything is an infinite parameter space.
Exactly. And like all people think mathematics, physics is the grand science of everything. And but when you think about it, for biology to exist, first of all, you need physical space. So all the laws in physics have to be, there. Then you
Yeah.
have all molecules, elements, so chemistry. And at the end, you have life that we have no idea how this thing emerges, and
Hmm.
it's biology. So everything that is contained in these other sciences, physics,
Hmm.
chemistry, is in biology, and you have
Yeah.
the basically variable life on top of that. So it's
Yeah.
really really interesting.
And once you add
Um,
consciousness and the brain to it, then when you start sort of self-referentially ponder how does that entire thing use all that knowledge to understand itself, it really becomes this circular philosophical bombshell that can really break your brain if you think about it too hard.
yeah, but it's cool, right? This is things that keeps us awake, so to say. So
Absolutely,
yeah,
absolutely.
and it
That's the
inspired
reason we're doing
also.
it.
Yeah, inspires a lot of like younger generations. I mean, I
I
was
hope
one
so.
of the things that as I said, one of the main motivation was understanding memory, and as I think memory is kind of a basis of consciousness, if you wish, in terms of human, and therefore I kind of entered this field. But let's go
Yeah.
back to your book. So now you're my guest here, so let's talk about your book. How did you find a motivation to write a book on first place?
Well, it's very simple. I at one point I was stuck in Russia, waiting for my visa to renew for 2 months, and I had nothing to do. And that's where the ball started rolling. So this friend that who I already mentioned, whose book I illustrated, she was active in popular science in Russia at that time. So she suggested linking me up with some people who were publishing popular science. So I started there. I started with some magazine articles, online blogs, and gradually it developed into a side job and that I was doing for a few years. And I developed some name recognition in Russia, and some audience, social media. And then it just became boring for me to compress everything into those, you know, well, in Russia we count characters, we don't count words. I don't know how many words I was writing, but it was always 10 to 12,000 characters. What is it? I don't know,
Mm,
3,000, 2,000 words, something like that. And it
something like
just
that.
became too restrictive. At some point, it got boring to me to have to start from the very beginning and then run out of space by the end of that article. It just sets a limit to how deep are the things that you can talk about. And I want to talk about something bigger. I wanted to talk about how I understood life and the human mind together, because I think you need to see the entire picture to understand each detail. You first have to at least glimpse the entirety of nature and your mind within it, and then you can zoom in on specific parts and get more interested in that. So what I wanted was to deliver the complete story in as compressed of a form as possible. And that's what this book became. It's a lot less compressed than the Russian version. By the time I got to the English version, it compressed even further. So now I really think it's it's fairly bite-sized for the really the enormity of what the book tries to cover.
It's really like enormity, as you said. It's you start from the origin of the universe and you end at a completely different place, complex conscious experiences and
Yeah.
interactions with humans. It is really, really, really fascinating. Yeah, I actually haven't mentioned that your book was already awarded several awards in Russia, so it was published already there, and now it's coming in English and
Yep.
in the rest of
Yep.
the world, so to say.
Yep.
And yeah,
But it's
it
become
is
a different
very.
book in the process too. It has been published in Russia, and like you said, still doing pretty well in there. But I have changed. A lot of new sciences come out between those two points in time. So it's really not just a translation of a book. It's a new book rewritten
Hmm.
by me as a new person, and in a different culture, in a different language. It was a very interesting
Hmm.
process to translate it, and I think the things that I understood about the difference between Russian and English at the outset were different from how it ended up being. I've learned a lot about America and the English language and just perceiving the world from that direction as opposed to perceiving it from the Russian direction.
Yeah, yeah, completely different cultures. I am pretty sure, as a person who is coming from Southeastern Europe,
Hmm.
and
Hmm.
yeah, I think I understand what you mean by saying that. Where
Yeah.
does the title come from? One
Hmm.
hand clapping.
So the
And of
title
course, I need to
refers
say the subtitle is Unraveling the Mysteries of the Human Mind, right? But one
Unraveling the
hand
Mystery of the
clapping, it's
Human
very
Mind.
interesting. Yeah, yeah,
Yeah,
yeah.
yeah. So the title comes from what's called a Zen koan. A koan is this type of mental exercise, this riddle. Sometimes it's described as a riddle without a solution. It's not supposed to be easily resolvable with a specific answer, but it's something that you ponder, some impossible twisty question that you ponder and meditate on, and by doing that you take your mind to a place where words alone can't take you. That's what a Zen koan is. And this specific Zen koan is inviting us to ponder something that common sense tells us doesn't exist. This sound of only one hand clapping. We think that there need to be two hands to come together and make the sound, and it invites us to imagine the sound of a single hand clapping. And I think that what the koan is trying to do is very similar to what we have to do if we want to understand consciousness. It would be nice if consciousness was about some scientific discovery that just hasn't been made yet, that one day we'll get an email that will say scientists have figured out what consciousness is. It's this physical force that was remained secret up to this point, and now we found it, and that's what it was all along. It would be nice, but it won't happen like this. What understanding consciousness requires is not some secret. It is a reframing of the entire question. It's twisting your mind into rethinking what is it? What is this experience that you are experiencing, and how does it fit into the cause and effect of the world at large? That's what you need to do to understand what consciousness is. And I think that's what a Zen koan is trying to get you to do as well. So this koan is for me a good metaphor for how to think about human consciousness and human life in general.
Speaking of human consciousness, what is your favorite theory? We have like so many of them. The integrated information theory from Tononi. You have Carl
Yeah.
Friston. You have Hameroff and Penrose proposing something that, hey, consciousness is actually somehow in the microtubulus in
Microtubules.
neurons. Yeah.
Yeah.
Or I don't know, there are panpsychism and dualism. And even, I don't know if you're familiar with this particular scientist, Donald Hoffman. He's in West Coast, and his claim is completely different, claiming that consciousness is actually the first, and then out of consciousness the physical world emerged, which is bonkers, but it's actually very interesting in our time of information and simulation theories. It's very interesting perspective. What is your favorite take on this hundreds years old question, even
Yeah.
millennia old question?
Yeah. Yeah, of course, it's a very difficult question, and I think that before we settle on a unified theory of consciousness, there'll be a lot of renaming and merging and unmerging of those theories, and eventually they will probably arrive at something in the middle. If I had to pick my favorite, I would say that I am in the camp of Carl Friston and
Mm.
Andy Clark, treating consciousness as as an innate feature of our brain that is centered on prediction, that consciousness arises as a result of our brains constantly predicting and explaining ongoing experience. This ongoing experience brings in new information into the brain, and the brain is trying to wrangle this new information into submission by assigning predetermined abstractions and patterns that had already extracted from it in the past, and by assigning those patterns onto ongoing information, modifying it in the process, and then having new information being assessed in this way. So this is the circular process of information being predicted, these predictions shaping ongoing information shaping predictions, and then vice versa, and in a circle, and that circle, that is what we experience as a consciousness. So in very simple terms, in a nutshell, that's what I believe it is. I also think that what I just described is very compatible with integrated information theory. It's just that integrated information theory, as it's currently formulated, I think makes some more specific predictions about the role of different parts of the brain, the prefrontal cortex or the parietal cortex, which may or may not prove true. I think that that's a little bit far away from my field of study for me to make informed judgment about the specifics of the predictions of integrated information theory and the global workspace theory as well. There was this recent study which had pre-assigned goals for what will decide if one theory is right and what theory is wrong, and they did those experiments, and basically it's still unclear. We we wanted to clarify which one is going to win and which one will lose, and well, both of them lost in some way. Both of them lost one in some way, lost in some way. So I sort of feel like that's where everything is. I do believe that consciousness is a feature of matter. I don't believe that there is something that we need to fundamentally discover, like the quantum effects in microtubules. To be honest, I don't want to mess with the microtubules people. Microtubule people seem to be very aggressive in my experience, and they will defend that theory to their death. So I don't want to fight with that. If it is microtubules, I'm happy with microtubules, but that's not where I go to naturally.
Yeah. By the way, microtubules, for people who don't know, these are these very interesting filaments, if you wish, scaffold that formed in the cells. One of the scaffold molecules, with neurofilaments and actin filaments. And people believe that in this they form like a tube, and in this tube somehow consciousness is somehow there. And one of the presumption was because of anesthesiological treatment lead to some kind of collapse of this microtubule. I'm not familiar as well enough to comment on that, but I have never met anybody who is defending this, so I can drop it here.
I've met them online, and they always defend those microtubules with a lot of force. But, I mean, to be honest, microtubules are extremely fascinating cellular organelles. Don't get me wrong; I love every component of the cell. It's just that, as far as I have seen it, I haven't seen enough evidence to say that microtubules have some unique properties that would explain something that other parts of the cell might not explain. I think that if you looked at the ribosome or a proteasome closely enough, you would also find some quantum effects in those. You would also find storage of information, degrees of freedom that are unexpected, and I think all of that is true. All parts of our cell are extremely complicated and are probably capable of doing more than we give them credit. That's true for microtubules. That's probably also true for many other parts of the cell. But that, to me, does not address the specific properties of consciousness necessarily.
So you mentioned that we might need to change our understanding of how we approach the problem. Do you think that currently we have the technology to address this problem of consciousness, or we're still not there yet?
Well, we can start changing our minds about it with the technology that we have. But of course, it would be nice to gain uh, a deeper control, maybe not even control, but a way to monitor what is happening in the human brain to a higher degree of precision than we can currently do. Because what limits us in many cases is that we can access individual neurons in research animals. But it's very hard to ask a research animal what's going on in their consciousness and what they're experiencing.
Mhm.
And we can't really get to that level of resolution and control in a human brain. So, of course, if our technology progressed to the point when we could know about molecular processes inside individual neurons, I'm sure that would advance our understanding of consciousness to a greater extent that our technology allows us today. But that being said, I think we already know enough about how the brain works um, to be able to assign mental experiences, mental functions to specific processes in our brain. Just need to be maybe more courageous about doing that. I think that neuroscientists are kind of fearful. They're scared of saying that that neuron is fear, and then this one is pleasure, and that this theta rhythm represents attention. It's a leap. It was always going to be a leap.
Mhm.
But I think we need to make it at one point or another if
Mhm.
we are to talk about consciousness in materialistic terms.
Mhm. Mhm. I mean, yeah, you have indeed. It's hard to study the human brain as invasively as in other animals, but you still have the option to actually poke it a little bit during, let's say, some extreme conditions when some patients need to be treated, and some kind of a
Yeah.
brain region need to be dissected out or something. Our previous guest actually participated in a similar study, and they were looking at neurons. Of course, now I forgot exactly which one, but it was about how people think about others, that they are thinking of something, or it's kind of
Like
crazy,
mirror
some theory
neurons.
of mind. No, it was not the mirror neurons, it was something else in the prefrontal cortex, but
Mm.
it was very interesting that actually you can design this experiment to kind of perform it with humans and pinpoint exactly neurons that have, as you said, particular task to do something. This kind of 300 neurons or whatever they have this task of doing that thing, and then they just found it out.
Yeah.
I'm really
It's
now mad at myself that I don't remember exactly which neurons were these, but it was a crazy study. It was published in Science, I think, recently. Yeah.
I think any time when we can relate a higher order aspect of human brain functioning to something really, really big, to something really, really small, like an individual molecule or receptor or a cell in the brain, that is fascinating.
Mhm,
And it's
mhm,
really rare. It's really, really
mhm,
rare to have that. So, Wilder Penfold studies where, like you said, you poke different parts of the cortex and you elicit these, a sensation of deja vu or color blue or anything that can go through your brain, you can elicit by poking some part of the brain. I mean, that is incredible, and that's really our
mhm,
foundation for how we understand the cerebral cortex now.
mhm,
Or psychedelic drugs, honestly. That's another any kind of psychotropic substance that predictably and specifically modifies how your brain perceives a certain aspect of reality. That is also
mhm,
fascinating.
mhm,
Or another technique that's used in neuroscience, binocular rivalry,
mhm,
where you can put on goggles that will deliver two different images into your eyes that are not compatible with each other. You can't make a three-dimensional percept out of them. And so your brain will end up constantly, endlessly switching between the two. It won't stop. You can't control it. It will go. You see one thing, and then a few seconds later you see another thing, and then the first thing, the other thing, and so on endlessly. So it all feels like in instances like that, you just feel how fragile and how material your mind is, that you can tweak a couple of molecules and you stick a wrench into this process that we don't give a second thought to, that just happens naturally. But there are ways that you can just tweak it a little bit and see that the whole thing falls apart. And that to me is some of the most valuable lessons from neuroscience that we'll hopefully have more of in the future.
I hope too as well that we are doing this work, of course, partially that because we want to understand principles, but also because we want to help people who actually need the help. At least I think this is the case
Of course.
when I'm doing science. Yeah. I think now it's a great time point to finish off our discussion, and I'd like to ask you, what take-home message do you want to convey to people who are reading One Hand Clapping, or who are going to read it actually, because it's still not there yet?
Yeah. Well, I think that when you consider your mind and your life within this greater context of life on Earth, which is what the book is doing, you contemplate your purpose and your meaning. And that's the thing, this is the philosophical things that people have been grappling with for thousands of years. What is my purpose and what does my life mean? And I think what seeing your life in the context of, evolution of life on Earth teaches you is that there is no purpose. Life is not looking to move anywhere in particular, but there is meaning. There's meaning in every single element of your body, your brain, your mind. There is a story behind every single aspect of your life that goes back eons, billions of years into the past, and connects everything to everything else. And if you trace each of those stories to that foundation, that is how you understand it. So that to me is worth living for. There might not be a purpose in life on Earth in general and in our individual lives specifically, but there is meaning in everything. And if you just focus on the meaningfulness rather than the purposelessness, I think you can be a happier person.
That's a great message, and I think it's a perfect ending of of our discussion.
Great.
Thank you very much for your time and for the valuable insights on memory
Thank you so
and
much. It's been
topics
a
such
pleasure.
as consciousness. Yeah. Thank you very much.